All right, thank you all for coming today for attending this short presentation focusing on improving soil health with sod-based rotation and cattle grazing. I'm Gabriel, I'm an assistant professor in soil water sciences. I'd like to acknowledge the contribution that Micah James, a PhD student in my lab, has made to this presentation. So when we talk about soil health, we are talking about a variety of things. It's a concept that has gained popularity in recent years. There's several definitions. Here's one of them. The one that we probably are more or most familiar with would be the one from the NRCS, which is a continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. And it kind of goes back to some extent to sustainability and the capacity of soils to just remain productive for our own purpose and for the purpose of ecosystems as well. Here's another good definition. And, when we're talking about soil health and how do we define and study it, we typically focus on all aspects of soil science and soil functioning. So that would be both or all of physical, chemical, and biological health. And here you have a variety of um, indicators and properties that are key for this. So why does it matter to have healthy soil? Or try to prevent a variety of issues. This is one of them being, soil erosion. That can be a big issue, as we are all aware. Other issues could be lack of soil fertility, lack of microbial activity, etc. So in this case, for this short presentation and for Micah's PhD dissertation, we're focused on two study sites in northwestern Florida. On the left-hand side, we have the grazing trial that Joseph Bay is maintaining, a mix of Bahia grass and a rhizoma peanut. So you see those two probes here. You see the rhizoma peanut here growing in between. And on the right-hand side here, we have cotton plants that are growing the sod-based rotation to Quincy that David Wright and Jim Marois in the past have maintained for a long period of time. So in these plots, we go out, we collect a variety of soils with a variety of instruments. So here you have two kind of more traditional push probes, something that of bigger volume. We have here a soil auger, and you can see the kind of soil that we're capable of getting. Here we have a push probe with a soil profile, so you can see different colors that represent different horizons. And then, for this specific project, and even though we're not going to be talking about this today, we may be using something like a geoprobe. Here's Micah, basically. And what this allows us to do is to go deeper into the soil profile to basically see things like, carbon accumulation, maybe nitrate leaching, etc. So one of the key issues when we try to sample for soil health, especially when we have animals, of course, is that these animals are not they result in heterogeneity. Right here we see cattle sitting in the shade, as most of us would do on a hot summer day. But this then is going to affect how these cattle excrete manure, excrete urine, etc., and therefore will affect our properties. Right? So if we have here this little shade structure, a little watering hole, how do we sample to make sure that we represent adequately the contribution of cattle and where they spend most of their time? That's one thing that's important when we quantify soil health or any other soil property, as a matter of fact. And the other part, of course, is depth. So here we have, accumulation of organic matter on top. We have here an horizon that's relatively alluviated, and then we get more of that reddish color deeper in the soil. So depending on how deep we go, of course, we're going to get a different signal, and there's pros and cons to go deeper into the soil, because then we start mixing different horizons. But on the other hand, it gives us a more integrated picture of what's actually going on. So then we take our soils back to the lab, I should say. Big question is whether we sieve them or not, whether we dry them or not. Different, again, pros and cons of this. There's an example of a protocol. We take soils, we weigh it on the top, we shake it with some extraction solution at the bottom. Then we typically pipette this into microplates. This is what these pipettes would do. And then here we have, this is a protein assay. We can see here a standard curve. So that's telling us that very little protein, a lot of protein, and you see the color development. And we can match the color of this curve with our samples to tell us something about soil protein. So a few results here. This is PNM. This is potential net mineralization. On top, we have June. On the bottom, we have October. This is for the conventional. This is probably our most striking results from this metric, which basically tells us how much nitrogen does the soil release. And what we see is that we see actually that it's highest in the cotton one, at least in the early summer, which makes sense because it follows peanut. So we see that between cotton one and cotton two, you see a big drop in the contribution of the soil to nitrogen. And later in the growing season, we don't see too much difference between the cotton, but the peanut is highest. In terms of the sod-based rotation, which is maybe our more interest, we have a little bit more puzzling results that we're trying to figure out exactly what's going on. But we do see some increase between the Bahia 1 and Bahia 2, at least early in the growing season. Then an increase in the cotton, and then the peanut in this case seems to be the highest in both years. So these are the kind of things that try to, it takes a little bit of time to measure these things, but they tell us something about how the soil is functioning currently and how it can be sustained in the future. So relatively quick and dirty. I want to thank you all for your attention. If you have any questions, I'm sure you can find the contact information on this field day. Thank you.